Thank you. All right, well, this was a talk that was supposed to be given a little while back, but due to COVID, it was postponed, and actually in some ways, slightly a good thing, because a big report has come out. And so today I am going to cover work, a broad range of work, a review of work that was done over many, many years, 10+ years or more, that was recently compiled in a review, State of Knowledge of Deep Sea Corals in New Zealand region. And this review was led and championed by a colleague of mine from NIWA called Di Tracy, and helped out by another colleague, Freya Horsvarsdottir, I'm never going to pronounce it correctly, who was at the time working at the Department of Conservation. And so this really is a collaborative project of many, many, many people who've contributed all over many years and from lots of different institutions. So it was NIWA, Fisheries New Zealand, Department of Conservation, and a lot of other collaborators from across New Zealand and internationally who have contributed to this knowledge of the deep sea corals in the New Zealand region. So here's the title page of this State of Knowledge Deep Sea Corals in New Zealand, which came out this year. And you can download the whole report. There's a link there, and I can provide a link in the chat. And as I said, this was really championed by Di Tracy, who kind of corralled us all and kept us all on track. It started with a workshop that was held at the Department of Conservation in 2017, where lots of people presented all different aspects of the deep sea corals, and we talked about the gaps in our knowledge. And then over the last few years, Di has been pulling together all the information from that workshop and all the recent publications and compiling it into this very nicely written document that's written very much for a stakeholder, government, but also public interest. So here's a picture of myself and Di, who has encouraged me to get involved in all this work. It really started back in 2008 when we were at sea on the RV Tangaroa, down on the Macquarie Ridge. And Di was one of the participants in that voyage. And we collected a whole bunch of deep sea corals during that voyage and used cameras to look at them too. And then later that year in 2008, there was the International Deep Sea Coral Symposium held in Wellington, which Di was helping to organize. And so she slowly sucked me into working on deep sea corals. And my role really has been sort of supporting Di and providing sort of information around deep sea corals to provide a more holistic picture of our understanding of the environment and where these things live. But I'll present a review in this review, all the other aspects that were included in the review. Briefly, I'm no expert in biology or taxonomy, but I will try to do my best to explain those things. I apologize in advance to any of the people who did the work if I've misinterpreted or misunderstood how to explain that work. So I'll just put that out there. A little bit about me. I did my PhD at ANU many years ago. I then worked for a couple of years at Geoscience Australia as part of the Coastal CRC. And I did a lot of my work on the Coastal CRC in the Fitzroy Keppel Bay area of southern Queensland. So it's quite nice to come back to, come to Queensland, because my PhD was done on the Southern Great Barrier Reef. And then my work and the CRC was done onshore in the Fitzroy-Keppel Bay area. Then I spent the last 12.5 years working at NIWA. And for those of you who don't know what NIWA is, it's sort of the equivalent of CSIRO in New Zealand. So it's a government-owned research institute. And NIWA covers basically water, atmosphere, but also the old Fisheries Institute, the old Oceanographic Institute, and then a lot of freshwater stuff as well as climate. For the last year, I've been here at UQ. So slowly getting to know my way around this place, but I apologize that I really haven't met as many people as I would have liked to in my first year, slightly handicapped by the COVID situation. So please feel free to message me after the talk if you want to have a chat and talk about some of these topics further. As a marine scientist, I've spent a lot of my time in working in open ocean, deep water settings, and I've had multiple voyages around the Southwest Pacific, into the tropics, down to Antarctica, and sort of various places in between. But I would probably say the majority of my time has spent looking in the Southern Ocean and Antarctic region. So that's my areas of interest. So here's a picture of Di, looking a little bit younger. And I put this in because I wanted to show you the scale that some of these corals grow to. We're not just talking about small little cup corals. Some are very small, but some of them grow to these tree-sized structures on the seafloor. And so these deep sea corals provide a significant habitat in deep water benthic ecosystems. And for that reason, they're sort of one of the key species for the designation of vulnerable marine ecosystems in the oceans. And also behind you can see a lot of jars of other corals and things. So this is the National Invertebrate Collection in Canberra, where there's thousands and thousands of species. And normally when I'd be giving this talk to, or similar talk to students, I'd have some examples to show you of deep sea corals, especially the very beautiful gold corals that are quite shiny and nice, and the bamboo corals, which I'll show you some examples of. So where do we find deep sea corals? Well, they've got a global distribution. Unlike the tropical corals, which is sort of concentrated in a tropical band, tropics to subtropical band, deep sea corals are found all over the world. And here's a website, a NOAA website, where you can look at the species distribution of different corals around the world. And actually, I think this species distribution is really more determined by where we've sampled and where we've, reported the corals from. There's many areas of the world where we probably would have corals, but no one's really bothered to look at the depth of water where we find these corals. When we talk about deep sea corals, we actually are talking about corals that can live anywhere from, mesophotic systems right down to thousands of meters. So they're typically corals that don't have photosynthetic zooxanthellae. They're ones that feed by filter feeding off detritus coming down through the water column. And so they're not just deep. Some of them are sort of fairly shallow, but most of them typically are found sort of below the thermocline typically in the Southern Hemisphere, sort of Antarctic intermediate waters, which I'll talk a bit more about. So if we zoom in a little bit more on Australia and New Zealand, you can see that New Zealand's quite a hot spot for these deep sea corals and sponges. And I'll show you another map in a future slide, which shows that we actually have a lot more than that's shown on this map. But I find it really interesting that Australia doesn't have a lot of dots on this map. I bet there are a lot more that could be put on there, a lot more records that should be put on there that are just not shown there. Or maybe we're just so focused on tropical corals in Australia that we haven't really been looking at deeper depths for these deep sea corals. But a whole bunch of work has been done off Tasmania, south of Tasmania in the Southern Ocean, by people like Ron Thrasher at CSIRO, who's done, and Karen Miller, who was also based down in Tasmania and now is in Western Australia, who've looked at the seamounts of Tasmania. And so we know that they definitely have quite a lot of deep sea coral down there in these waters. There's also corals all along the Louisville Ridge, because we had a voyage out there, NIWA had a voyage out there and found a series of corals out there. So there's a lot of corals that are probably sitting in international waters as well that just haven't really been explored or recorded yet. So here's the distribution of corals from 1967, when they were first really kind of made aware that these things existed and that they were found all around New Zealand. There's actually a lot of them found within the fjords. They've got big deep sea coral gardens in the fjords at only like 10 meters depth, but it's very dark at that depth. Very little light getting down there because of the thermocline and the water masses in the fjords. So if you ever want to go and have an actual look at these deep sea corals in their environment, then you can go to Fiordland and look at the deep sea coral. You can kind of go down and have a look. They have a little observatory there. And then in 2017, this is a recent compilation of where all the corals have been found around New Zealand and the Ross Sea. And you can see that we now have a lot of observations, and New Zealand really is a hot spot for these deep sea corals. Mostly sitting around places like Chatham Rise here, off to the east of New Zealand, but also a significant concentration along the Kermadec Ridge on these volcanic seamounts out here. Also down on the Macquarie Ridge, going down to Macquarie Island, which is of course part of Australia. And then sort of dotted around these other plateaux like Campbell Plateau and the Northern Challenger Plateau and Lord Howe Rise up to the north. And you can see a whole series down. I'm wondering if you can actually even see my mouse, which I'm guessing no. And I can't seem to get it down. Oh, here we go. So just to recap, this is Chatham Rise out here, where the majority of corals have been found, Kermadec Ridge, Macquarie Ridge down to the south, Challenger and Lord Howe Rise, and then the Campbell Plateau. So as I said, a lot of this work has been led by NIWA and the National Invertebrate Collection at NIWA. This is the RV Tangaroa, which I went down to Macquarie Ridge, where I first saw deep sea corals live on the video, but also collected them as part of a broader team. It's a 70-meter ocean-going vessel, 15 meters high. We still had waves breaking over the top of us occasionally in the Southern Ocean. And so this is the vessel that I've done most of my voyages on, and I'm looking forward to going out on the Investigator and maybe even the Falcor in the near future. So how do we sample deep sea corals? Well, typically the traditional method was epibenthic sleds and rock dredges that brought up these deep sea corals. And so here you can see in the bottom picture some pinky stalks. You've got some various different organisms here, but mostly different types of deep here in amongst the rocks. And these are rocks from the Macquarie Ridge, from that voyage in 2008 and the second voyage in 2011. So these things are fairly destructive, and it sort of breaks my heart every time I see a lot of biology come up with the rocks and stuff. But it's been the traditional way of us knowing where these corals are, and also collecting specimens which aren't always in the greatest of shapes because they come up rather abused by the rock dredges. So in the last decade or so, there's been a shift towards more deep-towed imaging systems, basically underwater cameras. This is the NIWA one that was built in-house called Dredges, and it basically has a whole bunch of batteries and some very big lights and a fairly standard camera. And it has a cable that means that we can see what's going on the bridge. And these things now have been really replaced by much more high-tech stuff, but this is still kind of the workhorse for a lot of the work that NIWA does. And here's some images that were taken by the cameras on the Macquarie Ridge, showing the sponge and coral gardens that live on the volcanic seamounts in that region. So some really quite spectacular ecosystems in these regions on these ridges. And ideally we would, of course, now move and take ROVs and AUVs, because the problem with deep water cameras is you can visualize these things, but you can't sample them. The problem with dredges is you can sample them, but you can't see how it fits into the broader scheme, and also you tend to destroy them. So now, I don't know if many of you have been watching the Smith Ocean voyages that are going on the depths around the edges of the Great Barrier Reef right now, but they use a ROV called Sebastian, and they can go down and do both the video and then also use their little claws to break off pieces of some of these deep sea corals that they're finding for genetic work and proper identification, because some of these species are quite hard to identify without looking in detail at the microstructure. And also, of course, the genetic information is telling us a lot more about how these species are linked to each other, but also more about other parts of their life and their function. So right now, as I said, the RV Falcor is out there sampling the deeper depths of the Great Barrier Reef. And my student, Vicky, was lucky enough to get on there a few weeks ago, and she's now out there for the next 7 weeks doing this kind of sampling. So I'm living vicariously through her right now, watching, try not to be too distracted by these videos. So deep sea corals are typically found on seamounts. They're found at various points on the seamount, along the edges and on the top. Majority of corals require a steep, hard substrate to actually attach to. And the reason why seamounts are useful is that they need quite a lot of food and nutrients. And so these seamounts, even in the middle of oligotrophic gyres where there isn't much productivity, can actually generate their own little productivity zone where the seamount and the waters interact and cause vertical mixing and allow for increased productivity around these seamounts. So that's why we typically find them on these seamounts. Of course, there are a few corals that are also found in soft substrates as well. So in the New Zealand region, we have this hotspot for deep sea corals. And as well as having a lot of different samples of deep sea corals, we also have quite a lot of diversity. And so you can see here that there's 116 different coral species of Scleractinia, which is of course related to the tropical corals in the New Zealand EEZ, and then even more when you look at the Ross Sea and the high seas around. And so I won't talk you through the whole table, but just to show you that there is a very high diversity as well as a lot of different locations that they live. And there's also quite a lot of endemism of these species in the New Zealand region. And so this is a table that was put together as part of a biodiversity review by Dennis Gordon in 2009. And it's been slightly updated by people like Stephen Cairns from the Smithsonian to show you that we've got quite a lot of species that are still undescribed, but we know are endemic or need to be described to prove that. And so an estimate of also how many species we still don't really know. So there's still a lot of work to do on the taxonomy of these species, and not a lot of taxonomists or deep sea coral taxonomists around. So typically, before the deep sea coral taxonomists come and visit and spend weeks and months in the National Invertebrate Collection doing all the detailed identification, NIWA and the Department of Conservation and Ministry of Fisheries have put together a whole series of these initial identification guides that can be taken by scientists and observers out to sea to give an initial ID of the corals before they get brought back. And so a lot of our coral collection actually does come from fishing vessels, bycatch records. Most of the deep sea fishing vessels in New Zealand have observers on board, and they basically are there to check for things like the deep sea corals. And there are certain rules that means that they can get the fishing vessel to move on if they catch too much of these corals. And so I'll talk a bit more about that at the end of the talk on the management strategy. Age and growth rates. Well, I showed you that image of a very, very large bubblegum coral right at the beginning. These things are very long-lived. They grow very slowly and they get quite old. And so there's a whole series of work been done using a range of different approaches to try and get an understanding of how old these things are and how quickly they grow, as part of a sort of way to assess the risk of bottom trawling and other disturbance to these vulnerable marine ecosystems. And so I've been involved in some of this work using radiogenic isotopes mostly to assess the rate of growth of these different species. So we just published a paper in 2020 dating this black coral here in the image to show that it was almost 400 years old and grows at around 5 to 9 millimeters per year as a linear growth rate, and then radially, of course, very, very, very small growth rate per year. And sometimes they use growth counts, growth zone counts, and other in aquaria approaches to assess them, but mostly it's been done by radiogenic isotopes because the growth zone counts have to be validated, and often they've been found to not really be annual rings because these things are living deep in the ocean and they don't really see seasons because they're basically seeing a fairly constant environment most of the year round. There may be a little bit of seasonality with respect to their food sources in productivity. So that's how some of them have been found to have growth bands, annual growth bands. This is some of my work. I initially first started looking at deep sea corals from their mineralogy point of view. I'm a marine geologist by training. I've become more of a biogeochemist, undetermined oceanographer over time because I can't quite decide what I want to work on. But these corals have a very wide range of different mineralogy. So the Scleractinia, just like the ones from the Great Barrier Reef, are made of aragonite, which is a type of calcium carbonate. The Alissonaceae, which are the bamboo and bubblegum corals, are high magnesium calcite. And some, like the stylastrids, are bimineralic, whereas the black and gold corals are proteinaceous. And so why does it matter? So these different minerals are harder or easier to produce by the organisms, but also they're going to make them vulnerable to things like ocean acidification, which is what I was sort of interested in from my perspective on this work. So aragonite is more susceptible to dissolution under ocean acidification, but high magnesium calcite is also equally vulnerable to dissolution as well, whereas low magnesium calcite is fairly stable and should be less. So I guess the question is around biomineralic, polymineralic species, how plastic these species are and whether they can switch between different types of minerals and can that help them survive. Reproduction, we really know very little about the reproduction of deep sea corals. It's thought that they're mostly broadcast spawners based on some work on Scleractinia by Rianne Waller. They're thought to reach their age of sexual maturity around 10 to 20 years, but there is examples of brooders, and there is genetic data to sort of support a sexual reproduction for these organisms. This leads me on to genetic connectivity. So there's been some work, not a lot yet, some work done on the genetic connectivity of these particular species between different regions and between different seamouths within the same region, using a range of microsatellite loci and DNA sequencing approaches. And here's some recent papers that have come out taking this approach on a couple of these Scleractinia corals, Solenosmilia variabilis and Madrepora oculata. Both show genetic differences between the north, central, and south in the case of Madrepora oculata, and whereas others don't show so much genetic difference and show much more similarity, suggesting that there's more connectivity. And the results from this recent paper suggested that this connectivity is mostly driven by ocean currents, different ocean circulation, and the food sources in the region. So one of the reasons why Chatham Rise is such a hotspot for these deep sea corals is it's the subtropical front, and there's a lot of productivity over the top of the rise, and so therefore these corals are getting a lot of food coming down to them. So this is sort of just to dive in a little bit more into the habitat and these different regions in New Zealand. There are lots of different potential habitats in the deep sea of New Zealand. We've got these big rises, plateaus, seamounts, volcanic chains that are all providing suitable habitat. And you can see this mud percent map that I produced last year. Basically, we tend to see these corals where we don't see much mud. So the mud, it shows a strong correlation where we get lower mud. We tend to get these higher corals on the Kermadec Ridge down at the Macquarie and on the Campbell Plateau. And across the Chatham Rise, we actually get quite a lot of corals just because it's strong currents and the mud is washed away. So bathymetry is playing a really strong role on the coral distribution, but bathymetry is also controlling the ocean currents. And so here's some maps showing the ocean currents at the surface here. We have we have at the surface here these warm currents coming in from Australia along the Tasman front, but we also have cold waters coming in from the Southern Ocean, which is why Chatham Rise is such a hotspot, because it's the combination of these waters mixing. But actually really important for the deep sea corals, because a lot of them sit at 1,000 meters, which I'll show you in a few slides, is the circulation of these intermediate water masses. And again, some are coming in from the north and some are coming in from the south. But also the deeper ocean currents as well, coming in from Antarctica mostly, also will be influencing some of the deeper corals. So here's what the depth distribution of the Scleractinia in the region looks like. And you can see here that there's this peak at around 1,000 meters. So we've got the Solomosmilia variabilis, Enolapsamia rostrata, Madrepora oculata, all sitting really around that 1,000 meter, dominated by that 1,000 meter peak in occurrence. Interestingly, Goniacorella demersa, we get two peaks, and this top peak is really the dominance of the top of the Chatham Rise, which is around 400 meters. And a lot of these Goniacorella sit on the top of the Chatham Rise. Oculina is a shallower water one found around New Zealand. But in general, you see that if we combine all these habitat forming Scleractinia, they typically peak around 1,000 meters, but also at this 400 meters on the top of Chatham Rise. When we look at the habitat forming Allancinacea, these are the bamboo and bubblegum corals. We get, again, a very similar peak around 1,000 meters in all of these, and this is the total. And so this is likely telling us that this is where the main habitat is for these organisms. But we use this distribution. But also another question is this just a depth and gear sampling bias that we see these peaks at around 1,000 meters and we're not seeing anything much deeper? And so here's a graph showing, or a map showing where all the samples have been taken around New Zealand, both fisheries and research samples. Here's the number of samples by gear type. So is it selected by gear type? But also you can see that the majority of the stations have been in the top 1,200 meters. Thousands and thousands of samples, sites have been collected in the top 1,200 meters. And there's very, very few samples that have been collected at depth. And here's just it on a log scale to show you that tail that you can barely see on the other graph. So we need to keep that in mind when we're thinking about their depth distribution. Is it just a sampling bias? So what I did with the depth samples is we kind of resampled, trying to take into account all those different stations, total stations, and normalized it to the total stations. And potentially we should be seeing some samples some of these records a little bit deeper for both the Scleractinian and the Allancinacea. I also used the distribution of these corals to determine what their thresholds and environmental parameters and tolerances were. And so here we've got the species down the side, but versus depth, which I've just shown you. But we also plotted against temperature, the dissolved oxygen at these depths, and also the aragonite saturation state, because both the Scleractinian and Allancinacea have fairly similar dissolution vulnerability. And what we found is that, you know, they like temperatures greater than 4 degrees typically, based on these samples, that they prefer oxygen levels greater than 150 micromolar per kilogram. But they also like to have the majority of them sit in the greater than 1 aragonite saturation waters. So that just basically means if they died, they wouldn't dissolve because the water around them is saturated with respect to aragonite. But there was definitely some that were able to live and tolerate down to lower saturation, so undersaturated at around 0.8. But work by Ron Thrasher has shown that corals, deep sea corals that are found below the saturation horizon are typically much more fragile and don't have the same internal structures that are seen in other corals. So while they may be surviving down at these depths, they're probably not thriving, and it probably costs them a lot of energy to build their skeletons down at these depths. So this was our assessment from New Zealand. And actually, globally, there's been work done looking at Scleractinia from all around the oceans, showing that, again, the aragonite saturation state is one of the dominant controllers of the depth range of these Scleractinia. And so here you have the same different species, along with Lophilia petusa, which is not found in the New Zealand region, but is one of the most common in the North Atlantic. And pretty much most of them sit above the aragonite state of 1, with a few that seem to tolerate a little bit of undersaturation, like Solomosmilia variabilis, Madrepora oculata, and Enopsamnia rostrata. So the deep sea corals in the New Zealand region are showing similar trends to these global distributions. Interestingly, they also pointed out that temperature and depth are also a strong control, but aragonite saturation is intimately linked to temperature and depth. So how do you tease out these different environmental drivers? So the next thing we did was we basically developed some algorithms to estimate the saturation states in the New Zealand region. And then my colleague Owen Anderson used a couple of different types of benthic habitat modeling software, boosted regression trees and Maxent, to try and look at what is controlling these species distribution. So is it the currents, the dynamic ocean, how strong the currents are, what the bottom temperature is, what the primary productivity is from the surface, dissolved organic matter in the water column, tidal currents, temperature gradients at the surface, as well as at the bottom, whether it's a seamount region or not, and aragonite and calcite saturation, but also slope. So these bubbles basically show the dominance of these different environmental parameters in controlling the distribution of these particular species. And then for Scleractinia, this is all the Scleractinia combined, and then each of the different habitat forming ones below, and then Alcyonacea, and then some of these Bathypathes is a black coral, so is Leopathes. Primnoa is a type of sea pen. So they looked at a range of different coral species, and then used these to assess the habitat suitability around New Zealand. So here's the results for Solomosmilia variabilis, one of the Scleractinians, and you can see that the areas where we get a lot of these corals are where we would expect, to get lots. We get, for the Solomosmilia, which lives a little bit deeper than some of the other Scleractinians, it's this red area is around the edge of the Campbell Plateau, but also along these Kermadec ridges and Macquarie ridges. For stylastrids, it's very, very similar. Those are the sort of precious hard coral, red corals. And then we took the same modeling approach, but went forward into the future. And so my initial looking at the future forecasts for aragonite saturation states in the New Zealand region suggested that the saturation horizon is going to shift from around 1,000 meters up to around 500 meters by 2100. So what implications does that have for the potential future distribution of Scleractinia in the deep sea around New Zealand? So you can see that basically today there's a strong probability that they will sit in these orange and red areas, but these orange and red areas are basically pretty much confined to the Chatham Rise and a few other shallower regions on the Lord Howe Rise by 2100, based on this predicted change in the saturation horizon of aragonite. This was using the GFDL model from the IPCC, one of the biogeochemistry models. All the different models from the IPCC gave very different results, and we chose the GFDL one because it matched most closely with the modern carbonate conditions. Unfortunately, these biogeochemical models are really not as good as the main physical models for IPCC, and so there's still a lot of work to do, and they don't always do a good job of representing the modern, and so it's hard to necessarily trust what they're going to do in the future as a result. So as a result, we haven't really published this future forecast one yet. It's a report for the Ministry of Primary Industries, now the Ministry of Fisheries, but it shows you that the area of where we expect these corals is going to really shrink and decline by 2100. So with that in mind, the threats to deep sea corals have been assessed a number of times. These are protected species by the UN Convention of Threatened Species. Unfortunately, in the New Zealand region, despite all this amazing knowledge that's been collected in the last 10 years by many of my colleagues, we still don't really have enough information to really do a very good job of determining whether these species are threatened or not. At the moment, so far, I think one species, the Paragorgia, the bubblegum coral, is the only one that has sufficient data to be identified as a species at risk, and they have plans on how to improve its protection. So as I just highlighted, the main threats to these species are environmental change, temperature, ocean acidification, changes in stratification and productivity, but also oxygen changes at these intermediate water depths and deep water depths. But also we have a whole bunch of human threats like fishing, seafloor mining, those Gonicorella demosa. A lot of those corals actually are attached to phosphate nodules along the Chatham Rise that a company would like to mine, and it's been going through the courts for now more than 5 years as to trying to get access to mine them, but the deep sea corals are preventing that from happening based on the fact that they exist there and they're a protected species. So hopefully that will stay that way, but there's a lot of phosphate in those phosphate nodules, and New Zealand needs phosphate. So there's a lot of push from an economic perspective. There's also, we don't know what the threat of microplastics is, but we're starting to find microplastics down in these deep sea. So if these are filter feeders, will they start taking up these fine microplastics? We have no evidence yet, but it's certainly something we need to keep in mind. Bottom trawling has been assessed, and my colleagues Malcolm Clark and Ashley Rowden and others have spent a lot of time thinking about this, looking at this, going back and resurveying areas that were once fished that have now been closed, so seamounts that have been closed. And there's very little evidence of any recovery within the 5 to 15 year timeframe on graveyard seamounts from the Chatham Rise that have been resurveyed multiple times over that time period. Potentially, there is some evidence of some of the smaller stylasterids that may not have been completely removed in the first place because they don't grow up and big, they tend to sit closer to the seafloor. So it may be that despite being fragile, these small species may be some of the first to come back. So it kind of depends on whether there was remnant populations there or not. And also about the dispersal distances of the larvae and whether there are local seamounts around that can actually be used to recruit populations. Certainly on the Chatham Rise, there's a lot of seamounts around, so there should be sufficient connectivity to allow that. But I think it's still ongoing work, and they've just come back, I think a couple of weeks ago, from a recent voyage back to the graveyard seamounts to do another assessment. So it'll be interesting to see what the results of that are. In New Zealand, just to sort of cover off a few, how are they protecting these? There's a whole series of different legislation to protect deep sea corals. And ongoing legislation and plans and acts like the New Zealand Biodiversity Strategy and Action Plan that was revised in 2016 and is being revised again already. There's a plan to implement a series of representative networks, MPA networks. They're tightening up on fisheries bycatch and bottom trawling in certain areas. and they need to now keep in mind climate change and biodiversity as part of their any plans to do anything in the oceans. Some of these deep sea corals are protected as part of the Fiordland National Park, and also the Kermadec sanctuary is going to, well, hopefully one day be put in place. Certain amount of protections already available up there, but they're going to hopefully become a full MPA at some point. So here's a map of the marine protected areas around New Zealand. There are 17 benthic protected areas. You can see that they don't really completely overlap with where we find deep sea corals. Some of them are very deep, well beyond where we would maybe expect deep sea corals to live. And so maybe we need to rethink these. These were established in 2007 before a lot of this work was undertaken. So there's a push in the Biodiversity Act to reassess this. There's also 16, I think actually 17, I might have got that wrong, 17 seamounts that have been closed, both unfished ones that have never been fished, but also ones that were previously fished, so they can assess, recovery and changes through time. On the high seas, there's the SPRIFMO, that's which is the South Pacific Regional Fisheries Management Organization, I think, but someone correct me if I'm wrong. That has recently put out a report on how to protect areas of the high seas. And of course, within the Antarctic region, there's the CAMLAR efforts to protect VMEs around Antarctica. So there's certainly efforts in place, but they probably need to be significantly reassessed based on the new review of all the data. There has been one attempt to actually undertake in aquaria studies of these deep sea corals. One of the hard things about these deep sea corals is they undergo barotropic trauma when they come up from the deep ocean. So they have to be brought up very, very, very slowly to ensure that they survive the pressure change. But they managed to keep Solemusmelia variabilis alive for over a year and look at how it may be impacted by ocean acidification. But there's also a lot of gaps still to be addressed. And so these were identified in the workshop. And James mentioned right at the beginning in the announcements that, there was the recent paper by people from CBCS who are looking at why there isn't more transplanting going on to save some of these species. And this was a topic that was brought up in a publication in 2015 by Ron Thresher and colleagues in Tasmania. They basically did an assessment of stakeholders about what the potential costs and benefits were of undertaking a range of different biological engineering and regulatory approaches to protecting deep sea corals. So I don't think there was anything particularly that jumped out of this perspective and review, but it's certainly something that's being talked about and considered by the deep sea coral community.